Hi, hello, good evening. So this is Krishnaveni and we are talking about the story of life on earth, and that's biology. So very warm good evening to all those who are here. So today is Friday, so we'll be talking about a class 12 chapter. Yes, so we are here talking about the world of biotechnology. So we are talking about how to alter a DNA, how to work beyond the scope of development, right? So we're talking about a much higher power, so much stronger power which comes with a greater responsibility. So alright, so we are here. So lecture 1, we actually introduced what is biotechnology, what are the tools that we basically use for biotechnology, but we haven't completed much, right? So we were just talking slowly because it is a new concept for you to understand. It is not easy for you to visualize. So when I say I am talking about evolution, when I'm talking about animal kingdom or plant kingdom, still you can correlate with the figures that I show you, right? So when I say evolution, you can imagine from early man to the person you are right now. So that is evolution, right? But so in case of animal kingdom and plant kingdom, yes, I am good. Oh, okay, you're in school now and still you managed to text. So thank you. I am good, thank you. But okay, finish your school and then do attend my class later, all right? But please do like and share before that. Yes, thank you, Keshavan, on my behalf. Yes, so when we are talking about plant kingdom or animal kingdom, it is easy for us to correlate with the pictures. But when you are talking about the molecular level or genetic level, it is not easy to correlate, because I don't know how you will imagine. So all you can imagine is a double-stranded DNA helix, right? But your double-stranded DNA helix is not visible. It is not visible to your naked eye, and for you to visualize it, for you to understand it at the gene level, it is pretty complicated. Yes, Priyanka Singh, good evening. It's not good morning. So I guess you just woke up. So it's good evening. Naveen, yes, good evening, and thank you for joining my session now. Yes. So we have been talking so much about biotech. So today I will actually go from start. I will again give you a brief introduction about what is restriction enzyme and how we are going forward to the next step. So I have a lot of GIFs and pictures to show you how actually gel electrophoresis occurs for you, so that you can visualize and correlate. Yes, Surinder, so good evening and thank you for joining my session today. Okay, so here we go forward. So hope you're all ready to alter your DNA very soon. So we are in lecture 2 talking about the principles and process of biotechnology. Alright, yes, good evening and thank you for joining my class. Okay, so whenever you see me, you feel like it's morning. Alright, so that gives me a good compliment. So you feel fresh when you look at me. So anyway, so it's a good morning for you. So I'll also greet you good morning. Yes, Harini, good evening and thank you so much for joining my session. Alright, so here we go. So where were we? What were we talking about? So what are the topics for the evening? So did you actually know how biotech actually originated? It's a very beautiful story. It's a very interesting story actually. So it actually dated back to around 18th century, but it's a very interesting story. So I'll tell you the story in between so that you don't lose focus now. So since it's beginning, so all of you will remain focused, but in between when you're talking at the threshold of a concept, I will tell you a story so that you don't divert, so that you don't fall asleep and you come back quickly. Alright, yes. So we'll see what are the topics for the evening. So we are already started with a few topics. So I have introduced what is biotechnology, and together we have defined what is biotechnology. Yes, hello Shubham sir. Then tools of recombinant DNA technology. So how it is being done? So how are we going to work with our DNA? How are we going to alter your DNA? So that is what we are going to talk under tools. So what are the weapons that we have? What are the tools that we have? So we spoke about 3 tools, I guess. So first one was your restriction enzyme, followed by a cloning vector, and the third one is your competent host. So we haven't spoken about each of them in detail. We have just introduced or discussed your restriction enzyme. So today we'll go forward and look into the remaining 2. Alright, and the third one is how actually the process happens. So what is the process of recombinant DNA technology? So we'll look into that. Alright, so coming back to the principle of biotechnology. So what is this principle? So who gave birth to these 2 techniques? So the 2 core techniques of biotechnology which led us to the modern development. One is genetic engineering. So what happened in genetic engineering? We learned that we are able to alter the genetic material, that is your RNA and DNA. So that gave birth to one of the first, actually that put off one of the first platform for your entire biotechnology, right? So that is your genetic engineering. So hi Gargi, good evening. Yes, good evening. Alright, so that was the first thing that gave birth to your genetic engineering. That is our ability to work on DNA. That is our ability to alter your DNA. Alright, the next one is your bioprocess engineering, the ability to maintain a sterile environment when working with a particular organism's genome. So that is bioprocess engineering. Alright, yes, so we were here, so I explained all this and then we went forward to talk about the recombinant DNA technology. So what is the recombinant DNA technology? So going back to our old very same example, so what was the example that we were talking about? How to take a smart gene from monkey A and put it into monkey B, right? So what was the steps that I told you? You'll take out a gene of interest and put it inside a vehicle and together you'll put it inside the host B, right? So that is the process of recombinant DNA technology. So first you will have to identify your gene of interest. So which gene you want to take and put it into which host, right? So that is called as gene of interest. Just give me a minute. Alright, so that is called as your gene of interest, the one that you're looking for, right? So you're going to take that, put it in a vector and put that into another organism. So that we will call it as gene of interest, and short form I will refer to as GOI. Alright, first you will identify. So how will you identify? Your DNA is 2 meters long. So if you want to identify 170 base pairs, which is your gene of interest, how will you do? So you go for techniques like bioinformatics, sequence analysis, etc. First you will sequence the entire DNA. Right, so what do you mean by sequencing? You will read the base pairs and then you will find out, okay, so this is the gene and it is present here, so I want to take that. So you will identify which is your gene of interest. Alright, so that is the first step. The next step is you will take out your gene of interest from the entire chromosomal DNA, because that is what you want to know, that is what you want to clone in another organism. So you pick out that gene of interest from the rest of the chromosomal DNA. Alright, then you pack the gene of interest in a plasmid, so that is step 3, and then you insert the recombinant plasmid. So when you pack the DNA inside a plasmid, it is a new combination, right? So you call that as a recombinant plasmid. So now you insert the entire recombinant plasmid inside a host. So these are the process of recombinant DNA technology. This is in layman terms that I am explaining you. Alright, okay. Finally, once you have inserted the recombinant plasmid into the host, you will check whether it is getting properly expressed. You have taken the gene of interest from some other organism and you are putting it inside your host. What if it is failed to express? Then the whole process goes to waste, right? So the last step is the most important one. You check for the expression of the recombinant plasmid. Alright, so we'll talk about each of the steps, how each of them is done in detail. So that is the story of the remaining chapter. So identifying gene of interest, I told you, you do it with the help of bioinformatics or some other software tool, where your gene is already mapped and you know it is of a particular base pairs, and you'll also find out if it has a recognition sequence for a restriction enzyme. Yes? Is this much clear? So first we are going to the second step. So we are done with this step. It is an in silico work where you do not have to do a lab work, but you will have to do a work that is related to your computer and software. So that is done. So moving on to step 2 here. Okay, so now we are going to isolate your gene of interest from its chromosomal DNA. So remember, you are going to take out only a part of your gene, okay, from the entire chromosomal DNA from monkey A. So how are you going to do it? With the help of restriction enzyme, you are going to chop the DNA. So restriction enzymes are also known as molecular scissors. Alright, so you are going to chop off your DNA. So these restriction enzymes belong to a class of enzymes which are called as nucleases. So anything that ends in A, SC belongs to enzymes. Fine. So there are two types of nucleases here. One is exonuclease and one is endo. So exo is outside, so it will chop on the terminals. Endo is inside, it will chop at a particular position on your double-stranded DNA. Alright, so this was the introduction I gave you for restriction enzyme. So this is the picture I showed you. So look here, you have a DNA template. So it is a double-stranded one. So this is from 5' to 3', 3' to 5'. Alright, so you have an exonuclease activity here, meaning it's chopping off at the ends, and here you have an endonuclease activity, it is chopping off at a particular position. Yes, so now going forward, we'll talk about endonuclease. So see, So we are talking about EcoR1. So it's going to cut exactly at a particular sequence. Yes, it is chopping off. So once it is chopped off, it is going to produce sticky ends, right? So do you remember this? We were talking about your EcoR1, right? So the figure that I showed you in the last class is where the recombinant process used the enzyme as EcoR1. So what is EcoR1? So this is how you write. So this is a restriction enzyme from your E. coli. So this stands for your genus name, these two for your species name, R stands for the strain, and the Roman number stands for the order in which they were identified, right? So EcoR1 is one of the endonuclease that chops off at a particular recognition sequence. So what is the sequence? The sequence is GAA-TTC, right? Yes. So this is the restriction sequence. So what is the complementary here? So where it will chop exactly at these two portions on both the strands. So that is the GIF that is showing here. Alright, so this is one example of your endonuclease which is able to cut both the strands of your DNA at a particular recognition sequence. And this is the recognition sequence for your EcoR1. Alright, then going forward, so these are examples of some of your endonucleases. So endonucleases can produce two type of ends. So one is sticky ends. So when it produces a overhanging ends, it is called as sticky ends, but when it cuts right in the middle, it is called as blunt end. So if you look at this figure, again it's an example of EcoR1, right? So it will cut exactly here and here. So it leaves a sticky end. But there are also something called as HAE3, which is also a restriction enzyme, which will give you blunt end, meaning it will exactly cut at this position. So when it exactly cuts in the middle, it gives you blunt ends. Alright, so this gives you sticky ends, this gives you blunt ends. So Priyanka says, out of two strands, which one is the coding template from which the coding takes place? So now why are you worried about the coding here? So for what you want to code? You want to ask for transcription? Yes. So if you have two strands of DNA, this is 5' to 3' and this is 3' to 5'. So transcription is pulled forward by what? Your RNA polymerase, right? So how does your RNA polymerase work? Your RNA polymerase works from 5' to 3'. So it will go in this direction, that this strand is your template strand. Alright, is this clear? So this is in case of a transcription, but here we are not talking anything related to your transcription. We are directly talking about chopping your DNA, especially your gene of interest from your chromosomal DNA. Alright, so we have so many enzymes. So we have over 900 enzymes that have been isolated from almost 330 strains according to your NCERT data. But apart from that, we have a lot of other restriction enzymes as well. So these are some of the enzymes and the organism it is isolated from. So this is the source. So this is the recognition sequence and the arrow indicates where it will chop off. Alright, yes, and this is the nature of ends, whether it will be a sticky end or whether it will be a blunt end. Alright, so again going back to the previous slide and showing you this. So look properly, EcoR1 is trying to chop. So when it produces hanging ends, it is called as sticky ends. Alright, now going back to the next slide. So look here, you have two sequence and when it chops exactly in the middle, it is divided into two equal halves and such a kind of end is called as a blunt end. Alright, yes. So now going forward to the next slide. So after the action of restriction enzyme, what happens? Now, okay, your restriction enzyme has worked. Okay, you're chopping your gene of interest from your chromosomal DNA. Alright. So now what is the next step? Yes, it is in molecular biology. So since I was talking about your 5' and 3', you got confused, I guess. And also the figure there in restriction enzyme talked about your template stand and coding stand. So you might have got confused. Alright, that's okay. So once your gene of interest has been chopped from your chromosomal DNA, so what is the next step? So how will you confirm that your restriction enzyme has worked? How will you guarantee? How will you go forward to the other step? So every time you can't come to step number 1 and keep isolating DNA from monkey A, right? So that is again a very tedious process. But to ensure that your process, that is your first process, has happened properly, so what will you do? So how does it happen? So restriction digestion, so you call it as digestion because it is going to cut. So they are performed by incubating your purified DNA molecule with restriction enzymes. So how it basically happens is, yes, so you will first take out the DNA, the chromosomal DNA from the organism A. So this is how you will get the DNA. So don't imagine DNA as double helix. So when you do it practically, this is how it will look like. So these are called as vials. Okay, they are small tubes, fine. So in that you will have DNA. So you will not have 10 ml, 15 ml. So it will all be in micro, macro concentration. So this can be around 2 microliters, so on, so forth. Yes, then you will add your restriction enzyme. That will also be in very minute concentration. Yes, so now you will leave it undisturbed. So that is called as incubation. So you will leave it for quite some time. Okay, so maybe for 1 hour, and you heat it at 37 degrees so that you have proper restriction digestion happening. Yes, so this is how your restriction digestion is performed. So it is all not visible to your naked eye. Whether your restriction enzyme has chopped off your DNA or not, you do not know. So how do you confirm it? So that is why you run what is called as agarose gel electrophoresis to confirm if your restriction enzyme has actually worked on your DNA or not. Alright, so that is how you confirm. Also, when you are performing a restriction digestion, so the restriction digestion is to chop off your gene of interest from your chromosomal DNA. At the same time, you have also the second component, that is your vector. So your vector is a closed figure like this. Now, if you want to insert your gene of interest into the vector also has to be chopped off, right? So the vector should be ready like this to accommodate your gene of interest. So the vector also has to be chopped off. So both your gene of interest and your vector has to be chopped off with the same restriction enzyme so that it is compactable. Alright, so here also you will have the same restriction enzyme, meaning it will chop off at the same recognition sequence. Here also you will chop the gene of interest from the same restriction enzyme so that you will have the same recognition sequence. So as such, both the digestion should be compactable. So here I mean you use the same restriction enzyme. Alright, is this clear till here? So Adil Rashid, so it says future doctor, hi. So hello, so yes, you're new. So thank you for joining a session. So I'm here teaching class 12. So we are talking about biotechnology principles and process. So we are talking about how your restriction enzyme, how your recombinant principle is working. Alright, so guys, till this, is it clear? Yes, so you understood practically how restriction digestion happens. So when I write a sequence and explain, so that is your theoretical part, but this is how practically the approach is. Fine? Okay, so going forward to the next step. So now we have to understand so now we have to confirm, right, whether your restriction digestion has worked or not. So we are entering into the next technique, so that is your gel electrophoresis to see if your DNA is chopped. So this is the matrix on which you are going to run your DNA sample. So this is made of agarose. Alright, so this is already present, so that is a template, okay, on which you will cast your agarose. So agarose is basically a wheat, so you get it from your red algae, so that is available in the market as a powder. Fine, so you will mix that powder in a particular concentration of water, then you will mix it with ETBR. Fine, because at the end, if you want to see the bands of DNA, you need to mix it with ETBR so that it is visualized under UV light. Fine, yes. So now you will mix the agarose in a particular concentration of water along with ETBR and then put it on this cast. So after some time it will solidify and it will become a gel. So that is the first step. Yes. So you will also make depressions like this, that is called as wells or combs. So in this you will load your DNA sample. Alright. Yes, Vanakkam Aman. So you told it correctly, so that is Vanakkam. Yes, you're right. Alright, so this is how your agarose, this is how you make the gel first. So once it is solidified, you start loading your samples, right? So this is the first step. The next one, this is how your apparatus will look like. So you will put your casted gel here, the gel that you made in the previous slide, you will put it inside this case. So this case will contain buffer so that your gel does not go dry. Fine. So this is the cascade. So you will connect it with an electric field. So according to the concept of gel electrophoresis that we read last class, so you are going to apply electric field so that the negatively charged DNA molecules move towards the positively charged anode, right? Yes, ETBR is ethidium bromide. Yes. So it is an intercalating agent so that it is so that your DNA is visible under UV light. Fine. All right. Yes. So this is how the setup will look like, and you probably run it around 50 volt to 110 volt. You can run it. 15 minutes it will take for your DNA to run. Then, so this is the front view. Now looking at the side view, so here is your negative electrode. So you will put your gel, the casted gel, from this side to this side. So you will load your DNA sample here. So this is the negative side, and then this is the positive side. So your DNA molecules will move from your negative side to your positive side in the presence of an electric field. Right? Is that understood? So this is the side view. That is the front view I am showing you, and this is the side view. So this is how it will run. And you will have buffer fill. So this is your buffer. Alright. So gel electrophoresis, DNA separate hota hai. So which is the, so where does your heavier one stay? It stays right near the wells only, where you have loaded. Yes, you're right. Good, Aman. So you're thorough with this chapter. Alright. Fine. So finally, after your 15-20 minutes, after you have run the sample, so this is how it will look like. So the heavier one, so you have loaded the DNA samples here. Okay. So this is where you load. And if your sample is heavier, it will stay right here, and if it has been fragmented, it will move quickly. So now, if you want to identify what is the base pair of the base pair of each fragment, you make sure you run what is called as a ladder. So ladder is a predefined DNA fragment whose sizes are already known to us. Okay, so this is commercially available. So these sizes are marked. So now, if you get a band corresponding to this, say for this. So this is 15,000 base pairs. So you know this fragment is 15,000 base pairs. This is somewhere around 450 base pairs. If your gene of interest that you have digested with an enzyme has to be 450 base pairs, then this is what you're looking for. So now you will know, yes, your restriction enzyme has worked, and that's why you're getting fragments. Otherwise, you will not get fragments, right? Otherwise, it is a complete chromosomal DNA. So these are the fragments. So this ensures that your restriction enzyme has worked well. So this is how you confirm. So here is a GIF. So this is how it will start running. So initially you will load on top, and slowly as you give electric field, so this is how it starts moving. Right? Is the process of gel electrophoresis clear for all of you? Okay. So now going forward, now you have separated your DNA fragment. So how did you do? Once again, you mix the restriction digested DNA with ETBR. Okay. So you don't mix it with ETBR. The ETBR is already added with your gel. So that is how you make the gel. So after it is done, you load the DNA samples in the well. Once you have loaded, you apply an electric field and you let the setup run. So after that, you let it run and then you view the bands. But before that, understand, so DNA fragments are negatively charged, so they move towards the positively charged anode. So that is the point number 1. And the principle on which it separates is exclusively based on size alone. Alright? Then smaller fragment moves faster. Okay, so once you have understood your restriction enzyme, yes, it has worked, it has chopped off my DNA. Alright, so you have found your gene of interest. Now, how will you pick your gene of interest from the gel? So that is how you isolate your gene of interest or your fragmented DNA. Yes, so the separated bands of DNA are cut from the agarose gel. So you actually cut it, the actual process of cutting, and you extract that DNA from the gel piece. So that process is known as elution. So understand, first you have a monkey A. We are going to take the smart gene from monkey A and we are going to put it in monkey B. Alright, so monkey B isn't that smart enough. So to identify the smart gene in the chromosome of monkey A, so you did some analysis and you found out, okay, so that smart gene is here and it is within this base pair, say 450 base pairs. Alright, then you also found out the recognition sequence that is present. Fine. Now you went on to find a recognition enzyme or a restriction enzyme that will chop your gene of interest. So you have chopped your gene of interest, you have taken it out. Fine, but you have to ensure that you have chopped it properly, so you do gel electrophoresis. So in gel electrophoresis also you get a confirmatory result. Yes, your DNA of interest or a gene of interest has been chopped. So once you get the confirmation, now you take that gene from the gel. So that process is called as elution. Alright, yes. So now when you take out that DNA from eluted gel, so that is used in combining with your plasmid. So you have to put it inside a plasmid to put it into monkey B, right? So your plasmid is ready and it is waiting. So you need to extract this purified DNA. So how will you do? So here I have a picture. So literally you will cut the piece of gel which has your particular gene of interest. So with the help of ladder, with the help of reference, you know this is this much of base pair is your gene of interest. So now you have identified, you will cut the piece of gel because that has your gene of interest. Alright. So once you cut the gel out, so this is the process that you will follow. You put the gel slice inside, then you will add a binding buffer to remove all the agarose and ETBR. After that, you ensure your DNA binds to a particular column. Yes, then you wash it, then you spin it, you dry it. So these are various processes. You don't have to go much in detail. I am just showing you how it happens. So how are you going to take that piece of DNA out? Finally, you elute it outside and you get your purified DNA fragment. So the step only takes so much of time to explain, but imagine how much of efficiency, how much of accuracy you need when you're performing experiments like this. Even if something goes wrong, the minimum amount of DNA you have will also go off. So I'm telling you, it is not in ml, it is not in ml, it is in microliter or it is in nanogram. So it is that less. So you have to work very, very carefully, very, very precisely, otherwise the entire step will go wrong. Alright, so this is how you elute. So finally we have a purified DNA ready. So we are going to put this in a vector and the entire vector we are going to put it in a host B. Alright, yes. So going into the next step, so now we are going to pack our gene of interest into a plasmid. So we have chopped off, we have ensured that our restriction enzyme has worked. Yes. So now you are chopping off your vector also. So both chopping off should be done with the same enzyme. So that is called as compactability. Alright. So now you have your purified DNA in the previous step, right, from elution. So you are going to insert that here. So once you insert that, this becomes a recombinant plasmid. Yes, so this is the third step. Now, if I say a vector has to take up your DNA, what are the qualities of this vector? So how should be your ideal cloning vector? You cannot take any vector and put your gene of interest inside, right? So this should also have some criteria, some eligibilities, etc. So we are just next going to talk about the properties of a cloning vector. Alright, so till this our purified DNA is ready. Now we are going to talk about the plasmid and then we will talk about how to ligate both of them. Alright, so the first thing, what is a vehicle or what is a vector DNA? So the definition goes like this. So DNA used as a carrier for transferring a fragment or a piece of foreign DNA. So you just have a piece of DNA, right? So that is your gene of interest. To transfer that to a particular host. So here our host is monkey B. You need what is called as a vehicle DNA or a vector DNA. Alright, so that is called as a vector. So what can act as a vector? Your plasmids and bacteriophages can act as a vector. So plasmids are extra chromosomal DNA that is present inside your bacteria as such. So it is naturally present. What are bacteriophages? So they are viruses which are going to infect your bacteria. They can also act as a plasmid. They can act as brilliant plasmids because both your plasmids and your bacteriophage, they have the ability to replicate inside a host independent of the chromosomal DNA, right? So where is your plasmid present? Your plasmid is present within the bacteria. So your bacteria will also contain chromosomal DNA. It will also contain your plasmid DNA. But both of them replicates independent of each other. So that is the criteria that we need. Even bacteriophage. So bacteriophage is a virus infecting your bacteria. So if given inside the host cell, it will also replicate. So that is why these two are brilliantly used as a vehicle or vector DNA. Alright. So bacteriophage will have high number per cell. So in a cell, the number of bacteriophage or the virus infecting bacteria is very high. So that is what we say high copy number. The ability to represent itself, the ability to multiply itself is called as a copy number. Okay, so here we need high copy numbers because we need lot of copy numbers. Because these plasmids are in a liquid, we will not know. So you produce more of the plasmids so that even if one goes wrong, the other is right. Alright, so that is why copy number is very, very important. So plasmid sometimes can be 1 to 2 plasmid per cell. There are some plasmids which can be 15 to 100 copies per cell. So this refers to your copy number, the ability to multiply or replicate itself. So that is very, very important. So the first criteria your plasmid should have is a very high copy number. So such a kind of plasmid is what you take so that you reduce the number of errors in your process. Alright, so that is called as copy number. So moving forward, cloning vectors. So this is how your cloning vector will look like. So plasmids are double-stranded circular DNA. So if you cut this off, it will become a linear structure, but otherwise it is a circular structure. Fine. So this is a cloning vector, and the cloning vector is nothing but DNA, right? So it will also have sequence. It will also have promoter and so on. So we are going to talk about what each of these sequences signify. So this is one good example of your plasmid, that is your engineered plasmid. We have artificially made a plasmid. So natural plasmid will not have all the genes that you need. So according to your need, according to the demand of the process, we have engineered a plasmid. So PUG19 was the first plasmid that was ever made. Sorry, PUG19. So it is a name of a plasmid. So what does PUG mean? So P, small p stands for plasmid. UC stands for University of California. So that is where the first plasmid was engineered. So we call that as PUG18. Okay. And then the next one that is commonly used is PBr322. Again, P stands for plasmid and BR stands for the two persons who discovered or engineered this plasmid. Alright? Yes? So you have, so look at the plasmid. There are so many sequences, right? So the first thing is, okay, so you have so many sequences. We'll talk about each of the sequence as I am telling you. So this is how, imagine this is how your plasmid is, and you're going to cut this off with a particular restriction enzyme and you're going to insert your gene of interest. You can cut anywhere, but where you're going to cut makes all the difference. So what are the features of a cloning vector? The first one is the origin of replication. So when we were talking about your central dogma of life, we were talking about your origin of replication, right? So what is the origin of replication? For a DNA to replicate, they need an initiation sequence. So that initiation site here is your origin of replication. Gene engineering example. So you use a plasmid which has very high copy number. So it also depends whether you are going to use a bacterial plasmid or whether you are going to use a virus as a plasmid. So it depends. If you are going to use bacteria as your bacterial plasmids, then the copy number should be at least 15 to 25. So that is ideally preferred. Alright. Okay. So coming back here, the origin of replication. So that is needed for a particular foreign DNA to replicate within a host. So if at all your any sequence has an origin of replication, it can originate without any problem. Alright, so that is needed to initiate or start the process of replication. So that is the origin of replication. So origin of replication also indirectly affects your copy number. If you have a very strong origin of replication, for example, you have a sequence like this. So this is your double-stranded DNA. So this is 5 prime. So you have your ORI sequence here and you have your gene of interest here. If this ORI sequence can initiate a very strong process of replication, then your gene of interest or your plasmid will be produced in more copies, right? So it will indirectly affect your copy number. So that is your ORI sequence or your origin of replication. So our ideal cloning vector should definitely have an origin of replication. Alright? Yes? Fine. Okay, so let me rub this. Now you should have a selectable marker. So just like how we found out that your restriction enzyme has worked or not, we ran the gel electrophorus, right? Now to understand whether your recombinant DNA, that is your gene of interest, and your plasmid has joined together, how will you confirm? So each step needs a confirmation, right? So talking back again, to know if your restriction enzyme has chopped off your gene of interest, we are running your gel electrophoresis. So that is all done. Now to ensure if your recombinant gene, that is your gene of interest, has attached with a plasmid, so what is the confirmation that you will do? So that is, you will ensure it is being transformed, right? So you'll have to know whether your gene of interest has incorporated with the plasmid sequence. So for that you use what is called as a selection step. So how can we say that ORI starts strong replication? Okay. So according to the process of central dogma of life, replication is initiated within a particular sequence, and that sequence is called as the origin of replication. So that is present in all plasmids naturally, and that is also how your replication process is initiated. So initially you have the formation of a replication bubble. So within a replication bubble you have a particular sequence where your DNA polymerase will go and bind. So only to your ORI sequence, your DNA polymerase, the major enzyme which drives your replication process happens. So it binds to your ORI sequence. That enzyme binds to ORI sequence, and that is how your replication proceeds. So without ORI sequence, your DNA polymerase will not bind, and hence your replication will not take place. Alright. So that is why I say ORI denotes a very strong replication start. So this is Rohit sir. So thank you for joining our session. Yes, so thank you. Alright. So to ensure that your recombinant DNA has integrated with your plasmid, we do what is called as a selection process. But how do you select? For that you need what is called as a selectable marker. Alright. Yes, I am coming here. We are talking about what is the selection process. So to select in the later step, we need some genes. So that has also to be present in your plasmid. So those are called as selectable markers. So here you have what is called as transformation. So this is one new process to ensure if your piece of DNA is introduced into the bacterium. Okay, so introduced into the vector. So now what you are doing, you have your recombinant DNA, your gene of interest. Say your gene of interest is ready and it has been packed with your plasmid. Now you are inserting it into a bacterium to check if they both have joined together, right? So how will you check? So if you're going to put this inside a bacterium, you need to check. So if both of them have entered your bacteria, it is called transformation. The ability of a particular piece of DNA to enter into a bacterium, that is called as transformation. All right. So to select if your bacteria has received your transformed DNA, we use certain genes as markers, right? So they are genes encoding resistance to ampicillin, chloramphenin, so all your antibiotic genes. So we basically have 4 antibiotics, so ampicillin, chloramphenicol, tetracycline or kanamycin. So to put it in a very simple term, for example, if you are going to a new place, okay, so a place which you have never been to, so you look for landmarks, right? And that's how you locate a particular place. Even if a Swiggy fellow is coming to your place, so how do you tell the address? You give them some landmark, right? So similarly, to ensure if your recombinant plasmid has entered the bacteria, we look for some markers, right? So if that marker is there, we confirm, okay, so this has entered your bacteria. So it is such an analogy that we are using here. All right. So your normal E. coli cells, they are not resistant to any kind of your antibiotics. They do not carry any of these marker genes. Now going back to your previous slide. So look at this PBR332. If you look, you have what is called as AMP-R. So this particular gene is very resistant to ampicillin. So ampicillin is an antibiotic and this gene is resistant to antibiotic. Fine. So similarly you have TET-R. So this is tetracycline. Again this gene, this pink shaded region is resistant to tetracycline. Now if you insert your gene of interest here, so if you chop it with BAM-H1, that is your restriction enzyme, and you insert it in this portion and you put it inside the bacteria. Now if you grow the bacteria in a tetracycline medium, this will die, right? Because this resistant gene is lost. Similarly, if you take out your ampicillin gene and insert your gene of interest here and put it inside a bacteria and grow the bacteria on an ampicillin plate, this will die. So with the help of this antibiotic resistant gene, you check whether your plasmid has entered inside your bacteria or not. So if this gene is present, it is able to grow in ampicillin. If this gene is present, it is able to grow in tetracycline. But if you chop off this portion and insert your gene of interest, then the resistance is lost, right? So which means with either of these two, you can check whether your process has happened or not. So that is the entire concept that I am trying to explain. Alright? Fine. The third feature is your cloning sites. You can clone it wherever you want. Like I told you, you can either insert it in ampicillin place or in tetracycline place. If you insert it here, then it should not grow in tetracycline place, but it should grow in ampicillin plate, right? You are either inserting in this position or this position. If you insert it here, it should not grow in tetracycline, but it should grow in ampicillin. If you are inserting it here, it should not grow in ampicillin, but it should grow in tetracycline containing medium. So in the medium, you give the antibiotic. Okay, so if this gene is present, it will grow against the antibiotic. Alright. If your gene has not been integrated, if your gene of interest has not been integrated with your plasmid, then this plasmid will grow in both the mediums, right? Both your ampicillin medium as well as in your tetracycline medium. So that's how you find recombinants and non-recombinant ones. That is how you eliminate, or that is how you confirm. So this is a very important process in your recombinant DNA technology. Alright? Yes? Suppose. So here one antibiotic resistant gene is used to select the transformin and the other one is used to get it. Okay, so you want me to explain it once more? I guess I have an empty slide after this. Alright, so I will explain it here. So you have your plasmid here. So this is the plasmid. Okay, so plasmid or vector. Now here in this plasmid, you are supposed to have what is called as a ORI sequence, so that it can replicate. Fine. Now apart from this, you also should have what is called as ampicillin gene or tetracycline gene. So ampicillin and tetracycline are your antibiotics. Okay. These are genes which can code, which acts as a resistance against the antibiotic. These are antibiotic resistant genes. So tetracycline and ampicillin are antibiotics. So inside your plasmid, you have what is called as tetracycline resistant gene. Meaning if the bacteria is grown on a tetracycline medium, it can still survive. So what are antibiotics? Ultimately, we use antibiotics to kill your bacteria. But if it has a resistant gene, then it can grow against the antibiotic, right? So is the case with ampicillin. Alright. Now you have your gene of interest ready. Fine. So now you are going to insert this in this position. So when you insert that in that position, so let me change the color of my pen. So here you have your gene of interest. The rest is the same. So here you have your ORI sequence. Here you have your ampicillin sequence. Right? So instead of tetracycline position, you have inserted your gene of interest, right? So now this is called as your recombinant recombinant plasmid. But my question is, how are you sure that your gene of interest has integrated exactly at this position? So according to your knowledge, you have inserted it in place of your tetracycline gene. You have chopped this off with your molecular scissors and you have inserted your gene of interest. But how do you know it has been properly integrated? That's why you put it inside a bacteria. So you insert this inside a bacteria so that this replicates and you can find. So once you insert this inside a bacteria, your bacteria will replicate the plasmid. So that is why it has a ORI sequence. Now it will replicate. Now once your bacteria has replicated, you will grow it in two medium. One is a medium containing ampicillin and another is a medium containing tetracycline. If your gene of interest here has integrated properly, then your bacteria will not grow in tetracycline medium. because it does not have the resistance gene. Suppose if it grows here, it has the ampicillin gene, right? Is this understood? So this is how you will ensure that your gene of interest has been integrated. If it's not growing here, it means these are your transformed ones. Alright? If the gene of interest has not been inserted at all, if it is not entered at all, it will grow on both the mediums. So it is a type of selection procedure. But remember, this is a very tedious process because you have to grow your bacteria in two types of plates, then you have to select your recombinant. So it is a very tricky process. Instead, we can use a very simple process. Instead of going for antibiotic resistance gene, so what is the developed technique is, you engineer a plasmid which has ORI sequence. It can also have some antibiotic sequence, but we are not bothered. Instead, you have what is called as an enzyme. Okay, so when this enzyme is being replicated, when this enzyme is being produced, it will produce a colored product. Fine. So you insert a gene that is coding for an enzyme. So now you mix it with your gene of interest. Alright. So now if you are inserting your gene of interest in this position, the enzyme will not be coded. So hence you will not get a colored product. So your recombinant ones will not produce a colored product, instead it will produce a white colored product. So which means you can ensure your gene of interest has been integrated. So this enzyme that we commonly use is your beta-galactosidase. This produces a colored product. Alright. So this process of inactivating a particular gene and inserting your gene of interest is called as insertional inactivation. You are inserting something at the cost of inactivating a particular gene. So this is called as insertional inactivation. Alright, yes? Yes, exactly. So at that particular gene of resistance, you have the restriction enzymes, right? So you will chop off at that particular place. Alright, so is this understood? So quickly going forward. So the last one is the vectors for cloning genes in plants and animals. So you have particular vectors that have been engineered. So first one is your Agrobacterium tumefaciens. So it is a pathogen. So it can directly infect a lot of plants. So it can cause a normal cell to become a tumorous cell. So such a kind of plasmids have been modified to be used as a cloning vector. Alright. Similarly, retroviruses, they are basically viruses which can cause a normal cell in your animal to become cancerous. Fine. So that has also been modified. So it has actually been disarmed. The virus genetic material has been disarmed. Fine. Okay. So here I have a picture of your Agrobacterium tumefaciens, and this is how your TI plasmid will look. So as such, this is a very dangerous plasmid because it has virulent genes. But when you are going to use this for the process of recombinant DNA technology, you will skip off your virulent genes. You will remove this virulent genes and you will artificially engineer a plasmid. It has if it has no antibiotic resistance gene, then it will not grow in either of the antibiotics. Alright? Yes? Okay. So with this we will stop today, and next class I will continue with your competent host, because now you have ensured that your recombinant plasmid is ready. Now you are going to put it in a host, that is your monkey bee. But your monkey bee should be able to take up this DNA, right? So that particular process is called as competency. So how competent your host is and what are the process involved in it, we will talk about it in the next class. So the next class will be on Monday. So we will carry forward with your lecture 3 on Monday, right? So I have got the slides, but let's stop here. I guess you have been saturated with the concept of your antibiotic resistance gene. So we'll stop right here. So we'll talk about all this in the next class. So here we are in the end of the lecture. So if you've genuinely liked or understood something from this lecture, so please do like, share and subscribe. Alright, and also there is no mentee today. So I will let you know about the mentee tomorrow. So tomorrow again at 4 p.m. I'll be talking about your class 11 chapter, that is Animal Kingdom. Alright, yes, so Naveen is done for the day. So even I am done. So thank you. So thank you all for joining my lecture today. So have a nice day ahead. Bye.